Hello, and welcome to our Insights lecture series. My name is Joe Komsky, and I'm the Senior Director of Philanthropy at Sanford Burnham Prebys. Well, it's been a year of our kids attending school from home, a year of wearing masks, a year of staying 6 feet apart, a year where we've lost friends and family to the pandemic. But now, through the work of our scientists here at Sanford Burnham Prebys and others around the world, we know a lot more about COVID-19 than we did a year ago. We know more about how we get it, how it attacks our bodies, how to treat those who are sick, and we have developed vaccines to help protect against it. But for everything that we've learned about this virus, there is much that we still don't know. And even though we have vaccines, we can't let our guard down in protecting ourselves and our loved ones. This is not to scare you, but to remind you that our scientists continue to work hard to understand all there is to know about this disease. We're going to hear from 4 of our scientists today. Their work ranges from finding medicines to treat those who contract the virus, to studying the long-term effects of the disease, to preparing for other potential pandemics. Most of you know how this works by now, but in case you don't, here's the agenda. Each scientist will take 5 to 10 minutes to describe the work that they are doing, and we will take questions for the scientists immediately after their presentation, and then move on to the next speaker. If you have a question, please submit it via the Q&A button on the bottom of your screen, and we will present them to the panelists. After all of our speakers present, time permitting, we'll bring all of them back on to take more questions from you. First up is Dr. Sumit Chanda. Sumit is the director of the Pathogenesis program at Sanford Burnham Prebys. He is a renowned virologist and has spent many years developing innovative approaches to treat viruses such as HIV, Ebola, dengue fever, and now the current coronavirus, SARS-CoV-2. Sumit, please go ahead.
Thanks, Joe, and thanks for putting this together, and I really appreciate the invitation, the opportunity to speak to the audience here. I put together a couple of slides just to give the audience a little bit of perspective on where we are right now and where things could be going. One of the most common questions I get these days is how is this going to end? And I keep trying to emphasize to people that there's not going to be a VE Day type event where the world leaders will proclaim that we vanquished the virus. It will be a slow burnout, and I think it's very hard to really predict anything about this virus. But what we can do is take lessons learned from the last worldwide acute respiratory viral pandemic, which is the 1918 influenza virus. So most of you know influenza, the 1918 influenza killed about 500,000 people in the US. It actually was suppressed not because of vaccines. Vaccines didn't come onto the scene until the 1930s. It was thought to have just gone through enough of the population where it was no longer viable as a global pandemic. But what it did was transition from pandemic, which means multiple viral outbreaks simultaneously in multiple countries, to becoming endemic, where you have hotspots of flare-ups that then are extinguished and then pop up again, to becoming seasonal, which is what we have now with the flu. And every year we go in, get our flu shots, and learn to live with it. Just to remind you, the toll of influenza is not insignificant. We have somewhere between 30 to 50,000 deaths from the flu every year here in the US. So it is still a major unmet medical need, but not one where that stops us from seeing family, going to school, enjoying going out to restaurants or sporting events. The one thing that we also want to learn from flu is that it comes back in pandemic mode every so often. And what this graph here shows you are the influenza pandemics that we've had over the last century since 1918. So they haven't been as horrific as 1918 due to various reasons, but they have been substantial. Most recently was the 2009 swine origin influenza that most of you can remember. Thankfully, it was not as pathogenic as we anticipated. But nonetheless, it did represent a new strain of flu that we had limited immunity to. And most of us anticipate that something similar will happen with coronavirus, that either this strain of coronavirus will evolve enough where it becomes so different that the large majority of the world's population will not have any pre-existing immunity to it. Or another coronavirus. To remind you that this is the third coronavirus outbreak we've had this century. And the next slide illustrates that, as well as the preponderance of other pandemics that have happened over history. And what you can see really here is that as we progress into current day times, there is really a compression of the number of pandemics that happen year after year. There are a number of causes for this: air travel, population density, moving into habitats where the animals that carry these viruses are residing. But the old adage that those who don't learn from history are condemned to repeat it is especially true, I think, from this experience. It's incredibly important that we as a scientific and medical community and as a society hold our leaders' feet to the fire to make sure we are better prepared for the next pandemic. And the next pandemic is not an if, it's a when. It will happen. We don't know exactly what the virus will be, but we have a pretty good idea of the list of top contenders. And it's incredibly important that we move to what I would consider a war footing and treat this as an existential threat and a clear and present danger it is. Much like the country did after 9/11, where we set up the Department of Homeland Security and instituted policies as well as measures that made sure that such an event couldn't happen again. We need to do the same thing when it comes to global pandemics. From our lab, what we're working on is developing broad spectrum antivirals. And these are antivirals that don't just treat a single virus, but multiple different viruses. And we think that this can be a strategy to be able to mitigate the impact of the next global, emergent global pandemic, even if we don't know what it is.
Sumit, thank you so much. That's very helpful. Quick question for you. I know that you recently just had a paper published on some of your work. Can you talk about that a little bit?
Sure. So one of our major focuses in the lab over the last year was to try to find existing drugs that could battle SARS-CoV-2. The reason we decided to focus on existing drugs is the normal path to drug discovery takes somewhere between 5 to 15 years. And obviously in a pandemic, we didn't have that kind of time. So what we did was we worked with our colleagues over at Scripps Research Institute right across the street and screened an incredible library of 12,000 known drugs that they had put together for potential compounds that can mitigate the replication of SARS-CoV-2. And last July, we reported 21 of these known drugs could be potentially effective against SARS-CoV-2. We took one of those drugs called clofazimine. Now, this is a leprosy drug that's been around for decades. It's orally bioavailable, which means that you can take it as a pill. And we were able to show that in hamster models, we get incredible efficacy for that drug. So this is really important because all of the drugs that are available right now to battle SARS-CoV-2 require hospitalization. So remdesivir, the antibody cocktails that you've all likely heard of, require you to go to the hospital and get an IV infusion. If you can get a drug that you can take as a pill, you can get it to more patients and more quickly. And we all know the quicker you get an antiviral to the patient, the better the likely outcome is. So if this drug can show efficacy in human in phase 2, phase 3 clinical trials, we envision a scenario where if you do get a COVID positive test, your doctor can phone in a prescription to your CVS. You can start onboarding this drug, and it will. You don't need to proceed to a hospital to be able to get treatment. So it should help a lot more people. And the other nice thing is it's cheap. It's about $1.50 a pill. You know, it will help underserved communities in America and especially in the third world. None of us are safe until all of us are safe. And so I think this drug will really help balance some of the health inequities that we've seen during the pandemic.
Is this kind of broad spectrum antiviral, has that been done before?
So the broad spectrum, so the drug that we're working on is a pan-coronavirus antiviral. It has been done, but at a small scale. So remdesivir, which is the drug that's been approved to treat SARS-CoV-2, originally was an Ebola drug. So it works against Ebola and it works against SARS-CoV-2, but that was never pre-positioned as a broad spectrum, right? And so the antiviral landscape really consists of a mindset of one drug, one bug, where you have a drug for flu, you have a drug for HIV, you have a drug for hepatitis C, but no one has really gone forward and tried to develop something that works across multiple virus classes, right? So I think that this is a unique opportunity where we focus our energy on finding those kinds of drugs because they are most likely to work on an emergent virus. So no virus comes out of nowhere. It's always coming from descending from some other lineage of virus. And so there's a high likelihood that if it works on, say, SARS-CoV-2, and MERS and SARS-1, it will work on the next coronavirus. So we are very excited about doing this, and I think it's really prime time to be focusing on efforts such as that.
Thank you, Sumit. We really appreciate that. We will bring you back at the end for some more questions, but we want to move on now. Our next speaker is Dr. Evan Snyder. Evan is the director for the Center for Stem Cells and Regenerative Medicine here at Sanford Burnham Prebys. Importantly, Dr. Snyder is an MD, PhD, and he played a vital role on the front lines of our community's battle with COVID. Evan, we're excited to hear from you. Please go ahead.
So, good afternoon, everyone. It's a real pleasure to speak with you. And this slide, in addition, gives me an opportunity to introduce my colleague, Sandra Leibel. We are both neonatologists, which means we both work with lungs. We're both developmental biologists, and you might say, what's a developmental biologist doing hanging out with great virologists like Sumit? And I guess it's illustrated here. What we're able to do is make mini lungs in a dish to try to model both the disease in the lungs that COVID-19 causes, but also therapies against it. Now, I should say that Sandra is a real lung biologist. Most of you know that I'm a stem cell biologist and a neuroscientist, but as a neuroscientist, I'm able to say that Sandra's the brains behind the lungs. But it's not just Sandra and I, it's actually a whole team, let me see if that is involved, and we affectionately actually call it the lung team, that has devoted a lot of effort to studying this. Now, what is this? What do we mean by this? It's a very interesting system that has a lot of opportunities. So you start with a patient and you can take out blood cells or skin cells. And then, and this is a very important point, the patients that we get these cells from can be a whole array of racial groups, different sexes, different ages, different genetic predispositions. But once we take the cells from this patient, what we can do is treat it with a set of molecules. These are part of the tools of stem cell biologists, and actually push them back in time so that they act as if they were in the earliest stages of embryogenesis. And then we use nature's recipe to push them to become lungs. And these can be lungs in a dish. Now, we can also push them to become inflammatory cells like macrophages, which is going to be important for the cytokine storm that we see. We can push them to become vasculature, blood vessels, because we know that COVID-19 is a very vascular disease, and you'll hear about that. But now we can examine them. We can examine them after they've been infected, and we can even change the environment in which they're in to try to emulate toxins or various environments that they may be exposed if they have predisposing conditions. Now, because we've emulated development, we actually see these lungs grow in front of our eyes, and we can test them and mix them with various cell types, test drugs against them, try to understand how does the virus enter, what drugs may block that. Now, very interestingly, this is Sandra's very last stage of making them develop. And what she's doing is adding a drug that you may be familiar with called dexamethasone. It's a steroid. This is the one drug that powerfully works, we know, in patients with COVID-19. We've always thought about it as being anti-inflammatory, but it's kind of interesting too, that this is a developmentally important drug, which may mean that maybe some of the repair is actually intrinsic to self-repair and to stem cells in the lung. Now, I'm going to end here with a comment that Joe made, which was that I was able to make what we do at the bench kind of relevant in my real life, where during the holidays, I actually took care of COVID-19 patients. And I have to tell you, and we can talk more about that later on, that put in perspective the stuff we're doing in the lab when we actually had to confront patients where there is a huge degree of disease disparity. So I was happy we were studying that. And part of what we did was turn a gymnasium from a school into an intensive care unit. So thank you very much, and we'll be eager to take questions later on.
Evan, thank you so much. I do want to hear a little bit more about this, because this is really remarkable. You know, a lot of times people think that our scientists are just, you're in a lab, you're removed from the day-to-day of the suffering of disease. You weren't. You were there, and you were helping to take care of patients. Did you see things, did you observe things there that have sort of informed your research?
Yeah, first of all, you know, intellectually, we can talk about how this disease unfairly hits various groups in different socioeconomic groups. And as I said, we're studying these stem cells. They're called induced pluripotent cells taken from different groups. And we can study intellectually whether or not they are predisposed to infection more than other groups, whether they respond to drugs different than other groups, and maybe try to play with dosing or play with synergistic drugs. But actually seeing the patients made me realize how much of this disparity is due to disparity in socioeconomic conditions, including not only the fact that people are clustered together in small housing, but that they are predisposed to having a great deal of comorbidities, obesity, diabetes, hypertension, things of that sort. So I would say it invigorated me to go back and be even more assiduous in what we do at the bench to come up with drugs. And as we'll talk about, we're still going to need drugs even after we have the vaccines.
So you're able to recreate some of those conditions in the lab. We have a specific question here about people with COPD or asthma and how safe it is to take the vaccine. Have you learned anything about that where you would be able to answer that question?
I think both from my clinical experience and also from the lab experience, that I would say that those with COPD and asthma, when they do get the disease, are more severe in terms of their symptomatology. And what we used to do for all the patients is we always gave them bronchodilators. Even if they didn't have predisposing conditions with asthma, we always gave them steroids. We always gave them anticoagulants. So having these predisposing conditions make the disease worse. A vaccine does not exacerbate those conditions. And I would say that anything that can keep a patient with these predisposing conditions from acquiring the true full virus is a benefit. The vaccine itself will not exacerbate those conditions.
And through your research of the different groups, age groups, racial groups, things like that, what have you learned anything that we're able to continue to move forward in terms of helping some of those groups that have been hit hardest by this?
Yes, I think a lot of it is now. Sandra and I have some preliminary data, and it's exceeding preliminary, that when they are infected, when these organoids are infected, that maybe there's more infection with, let's say, Hispanic males than with Caucasian females or something like that. And we can compensate by playing with dose. But I would say that probably the most important lesson I learned is how important public health interventions are. Really trying to prevent the spread of the disease by providing opportunities for groups that don't have the luxury that many of us have to have really good PPE, to have really good physical distancing, and to have very effective quarantining. And I would say those are the groups that really need the vaccines, really need the vaccines.
Evan, thank you very much. Again, in the interest of time, we're going to continue to move on, but we will welcome you back at the end and we'll take more questions. So now I'd like to introduce Dr. Ann Bang. Ann joined Sanford Burnham Prebys in 2010 and is the director of cell biology for our Conrad Prebys Center for Chemical Genomics. Recently, she's been using stem cell models of brain and kidney to investigate how COVID-19 impacts these organs. Ann, please tell us what you're learning. Go ahead.
Great. All right, so like Evan, I'm a stem cell biologist, and in my lab, we develop human stem cell models of disease to use for drug discovery. So we're located in the Prebys Center, and the Prebys Center is a state-of-the-art drug discovery center. It's a really unique resource that we have at Sanford Burnham Prebys. So I'm going to pick up where Evan left off and give a great introduction and tell you about work that we've been doing using organoids we make from human stem cells to study COVID. So I think everyone knows that not only does COVID-19 have devastating effects on the lungs, but it also impacts other organ systems, and this includes the kidneys and the brain. So a main goal of our research right now is to identify which cells are affected by COVID-19 in these organs. So are these indirect effects, for instance, from the cytokine storms that cause a lot of inflammation or blood clots that cause loss of blood flow, or are they direct? Is COVID actually infecting cells in these organs? So it's important to know this because our goal is to be able to design targeted therapeutic approaches. And also we really want to understand what the long-term effects of infection are. 10% of patients right now have are long haulers. And so they have symptoms of the disease that last for months after the virus has been cleared. So it's really a problem. So I want to start out and talk about our work with brain organoids. So COVID-19 is associated with a really wide range of neurological problems. So this includes fatigue, headaches, loss of smell, and cognitive dysfunction or brain fog. So these are really serious problems. So together with my collaborators, Delatorre, a virologist at Scripps, and Ming and Song, who are also stem cell biologists at University of Pennsylvania, we systematically looked at multiple brain cell types and organoids, brain organoids made from stem cells, infected them with SARS-CoV-2. And these included neurons, astrocytes, microglia, which are immune cells that are in the brain. Now, interestingly, the only cell type where we saw significantly high levels of infection is a cell from a brain structure that's known as the choroid plexus, and that's shown here in green. So the choroid plexus plays really important barrier role. So it keeps, for instance, wayward immune cells from getting into the brain, or pathogens like viruses and bacteria. And it also produces cerebral spinal fluid. So this is the fluid in the canal of your brainstem that is taken when you get a lumbar puncture, right? So cerebral spinal fluid has lots of important functions. Your brain is basically floating in this fluid. So it maintains appropriate pressure of blood flow, clears out waste, and protects against injury, like when you jar the brain. So here are pictures of choroid plexus organoids. These were made in the Song Lab. And so here you can see in this organoid, choroid plexus cells, they have this interesting structure around the periphery. And when you look up close, these are ones that we've infected with the SARS-CoV-2 virus, which causes COVID-19. You can see that many of the cells are specifically infected. So this is a case where SARS-CoV-2 could be having a direct effect on these choroid plexus cells. Now this is work that Hao and Zhao in my lab did. She also went and used choroid plexus cells taken from human brains, so these are primary cells, and showed that they could also be infected with SARS-CoV-2. So the consequences of this infection are cell death, loss of this barrier function, and also increased secretion of cytokines. Some of these cytokines are ones that are actually found in spinal taps of the cerebral spinal fluid in COVID-19 patients. So we feel like we have the beginnings of a system to begin to study what's happening with COVID-19 and these neurological problems. Does it involve the choroid plexus? And can we think of ways to prevent these problems and treat them? Okay. So another serious problem that COVID-19 patients experience is kidney dysfunction and acute kidney injury, or AKI. So this is really common in patients, shockingly common actually, that are admitted to the hospital. So depending on the study you look at, it's up to 57% of COVID-19 patients that are hospitalized. And nearly 80% of those that end up in the ICU can have this acute kidney injury. And this can lead to increased risk for chronic kidney disease for a lifetime. So I've started this project again with collaborators, takes a village, Della Torre, virologist at Scripps, McKay, who's a nephrologist, so she's seen kidney patients with COVID-19, and Ulovich, who's an immunologist from Scripps. So what we've done is taken the same approach. And so in my lab, we've made kidney organoids from human-induced pluripotent stem cells. And here in this up-close picture, you can see these beautiful structures. The green are what are known as proximal tubules, and the red are podocytes. And together, what these structures do in the human kidney is that they filter waste out of the blood. So interestingly, when they look at kidneys from patients, from COVID-19 patients that have had kidney dysfunction, what they see is that the proximal tubule tissue is injured. And so what we've done now is we've taken these kidney organoids, we've infected them with SARS-CoV-2, and we are doing this experiment as we're speaking right now. So we don't have the answer yet, but we hope to have one soon. So our next step in this study is to take what we've learned and the cell systems that we've developed in brain and kidney organoid and try and get a better understanding of the long-term effects of infection and how to design targeted therapeutics, taking into account what we've learned to prevent and treat these problems that develop in long-haul patients. So I'll stop there, take any questions.
Anne, thank you so much. You know, this is really interesting. This research is really interesting because this is sort of the first, I don't know about the first, but this is the research we need to even know that things are going wrong in the brain and the kidneys. I think when things first started, everybody thought of it as, you know, it might be in your lungs. But this is really important research, isn't it? Yeah,
there's a lot of unknowns right now. And, you know, they're doing more analysis of autopsy organs that they take from patients that have died. There's still not enough information to know exactly what's going on. We know that they find virus in these tissues. We don't know how it got there, right? And again, whether or not the effects are indirect or direct. And it makes a big difference how this happens, about how you're going to treat it. You really need to know what exactly are the sequence of events that happen to design the right therapeutics. And these are serious problems that people are having. And it's going to have a very big impact considering the number of people who have been sick on our whole medical system in the years to come.
Is the next step for you in this research to look at drugs that might mitigate the effects of the disease?
Yes, definitely. And they're candidate drugs, for instance, the inflammatory molecules that are produced in these infected choroid plexus. Could we stop that? And there's already data that says that dexamethasone, which Dr. Snyder mentioned, is one approach. And as well, we want to take these and build more complex models, for instance, where we bring in blood vasculature, which is already affected.
Not to scare anybody, and obviously the long-term effects we're unaware of because we're not long-term yet, but if it seems to me if it's affecting these cells in the spinal fluid, in the brain, or like you said, our kidneys' ability to filter toxins out of the blood, that there could really be some long-term effects of people who were sick, have recovered, but there is still disease that's doing damage to their body. Yeah,
they're vulnerable now, right? And so I read about people who are recovered and then months later they start to have these symptoms. So yeah, it's definitely, I think, something that's going to be a problem going forward.
Question here from the audience about, do we find that the long haulers fall into certain age groups?
I do not know that answer, what the epidemiology around that is. I know that there are certainly examples of both young and older people, right? And, I think young people feel invulnerable and they're not, right? So I would imagine that it could be affected by your own genetics, right? Your own health status when you get sick, irrelevant of what your age is.
Okay. One of the really interesting things when we do these is many of our own scientists tune in, and one of them has a question: what are the receptors to which SARS-CoV-2 binds for each of the different organs?
In both cases, it's ACE2. So in the case of the choroid plexus, it's ACE2, and we showed that in our study. In the case of kidney, it's known that ACE2 is expressed on the proximal tubules, which is pretty provocative. But we don't know yet if those cells are directly infected or if it's a secondary effect of, for instance, from macrophage, and so we don't know that yet. But we feel like we're getting the system set up where we can actually study that.
And then one more question from the audience, sort of following up what we said. So people who are asymptomatic could have issues later on in life down the road.
Yes, definitely. And that's something else that I think that we're beginning to see. Yeah.
Okay. Anne, thank you so much. We really appreciate it. We will take more questions again as we go on. But now we are going to introduce Dr. Carl Ware. Carl is the director of Sanford Burnham Prebys' Infectious and Inflammatory Diseases Center. And as he will show you, Carl was on the front lines of the battle against COVID-19 in the testing of vaccines. He is currently testing an antibody to inhibit something called the cytokine storm, which occurs in patients with severe pneumonia from COVID-19. Carl, please tell us about your work.
Well, thank you very much, Joe. And let me just congratulate the audience for their interest in the science and also the questions about this pandemic. This is a real sign to us scientists that we can have an impact in the population and help sort of in this pandemic. So I just want to congratulate the audience for being here to welcome us on board. So you've heard from several of my colleagues that in the later stages of COVID-19, there's this inflammatory process by the immune system that normally will protect you, but in the case of some individuals, these at-risk groups, if you will, the immune system becomes dysregulated. It is overactive, and in that setting, it can cause tissue damage. That's part of its mechanism, if you will, in controlling pathogens is that it can kill cells and get rid of them. And so what we're seeing here on the left panel is a little cartoon that sort of describes my work as an immunologist with a focus on cytokines that are involved in inflammatory responses in the lungs and elsewhere. It's been part of my focus as an immunologist to study autoimmune diseases. And we've taken that knowledge now and turned it around and begun to look at the role of the cytokine storm in driving the progression of patients into this severe inflammatory process where they can't breathe. And in some individuals, they're destined to die. And so one of our goals is to understand these cytokines. And one of the cytokines that I worked on for many years in my career, we call it LIGHT. It's a long acronym for a protein that we call as part of the cytokine armament of the immune system. And what you can see on the right-hand panel is that in patients that are ventilated, on the right-hand side or in the middle that are non-ventilated, but they're in the hospital, their levels of this cytokine in their circulation is elevated 5 to 10-fold above normal levels. And that's a good biomarker to tell us that, in fact, there is something awry going on in these patients. And where we are currently at in this knowledge base of this particular cytokine is that an antibody that I developed a few years back for autoimmune diseases, we have now partnered with a biotech company and have this in a phase 2 clinical trial. And the initial results look very exciting in protecting people from dying and also shorten their hospital stay. So we're very excited by the initial results, but of course, we need to move this forward into a larger patient population. And that's one of the exciting things that I find in the work that we do around immunology and cytokines. And of course, the goal of all of us at multiple levels here, my colleagues, is to really figure out how to control the immune system in a way that provides protection without causing undue harm to the body. As Anne mentioned, there's a lot of side effect in the long haulers in their kidney and their brain. And what we're hoping is that by using drugs that alter the function of light, that we'll be able to mitigate those situations and prevent people from having all these problems associated with the long term, like kidney damage and neurologic damage, as well as other settings. So one of the things that we all know now that can help us fight this pandemic, let's see, trying to move to the next slide here. Oh, there we go, is to get the vaccine. It's as simple as that. And so back in mid last year, I volunteered for the Moderna trial, became a part of that clinical trial to really prove that the vaccine was safe and also protected us against the virus infection. Now, those volunteers, which numbered about 30,000 people of all racial groups, of all age groups and sex, participated in this study. That's a very large number of patients. And in all cases, there was no issue with safety. And the protection level was very, very high, 90%, 95% coverage. And in those individuals, that remaining 5%, they were all protected from serious, severe disease. And so I'm often asked, why did I volunteer? I mean, wasn't it risky? And, you know, it's not. For me as an immunologist, I really understand the science of vaccines. So I wanted to do my part in addition to the laboratory work that we do. And in that regards, you know, I went through and participated in this trial and over the last, a month ago, I got my vaccine, the real thing. And so I'm feeling like I'm pretty protected at this point. And, you know, the vaccine can protect you from infection, but it also helps block the spreading of the virus to other people. And I think that's a really important feature of why vaccines are our best hope. But again, like my colleagues, we also need therapies to help those individuals that progress beyond or even prevent the disease. And but there's really, the vaccine is the key here. And so one of the things I like to talk about is that once you get that vaccine, you really know that you're protected for at least the strains that we have now. And for me, I got that sense of euphoria of being liberated from this pandemic. And we call it the immune aura. This is not a throwback to the 60s or 70s. This is really one of the features of your immune system is that the antibodies that are produced that block the virus infection, they're found in every nook and cranny of your body. And in that regard, you're protected no matter how the virus gets to you. We also have that backup of having immune cells that can attack those virus infected cells and eliminate them before they become a production site for the vaccine. So with that, again, I'll thank you very much for your attention and answer your questions.
Carl, thank you so much. Real quick question from me in regards to the cytokine storm. Are there different sort of sizes or severities of that storm? Can it be smaller and there's less damage done? Can it be larger and there's more damage done?
Well, like the study that I showed there, the levels of the cytokine can be predictors of whether or not individuals will progress through to the very serious forms of respiratory distress. And so in that regard, the level of cytokines can have some predictive value in that regard.
So is this something where we could eventually determine some sort of test as to who might be more likely to manifest some of these diseases that we're talking about?
Yeah, it's still early, but we think that, for example, light, and there are a couple of other cytokines like interleukin-6. These are cytokines, again, with potential predictive capabilities. We could do and develop very rapid tests. It doesn't take much of these cytokines in your blood to actually have a major impact on the physiology. So the levels of protein that we're talking about are in the picogram level, which is far less than most of the other molecules that circulate in our plasma.
Carl, do we see any effect? Do you think that there's an effect, difference between children and adults who maybe were affected in this and things like that?
Well, we haven't seen much of this, what we call the cytokine storm in children. As you know, there are certainly conditions in which some children suffer very severe responses to the coronavirus infection. In adults, I think this is a key issue to resolve and to understand how it may contribute to disease.
We have a question from the audience. Was the initial test vaccine different from the other vaccine that you got recently? And do you have extra protection because you got two? And I know you...
Great question. As it turns out, the volunteer study that I was involved in, I was in the placebo group. So I maintained my sensitivity to infection. However, I have completed the study with the Pfizer vaccine. So.
Very nice. Carl, thank you. We're going to bring everybody back on now. We have lots of questions, and as people come back on, Carl, we'll start with you. Just people who are asking, they've received the vaccine, but they want to go see family who maybe hasn't received the vaccine yet. Maybe they want to fly on a plane to get there. The people that they want to see are different age groups. Sometimes they want to see their children who might be in their 50s, maybe their grandchildren who are in their 20s. Is all of that safe to do? And what other precautions should people take when they're out trying to see folks like that?
Well, 6 months ago, I think we all felt that the vaccine would protect us, but as Sumit indicated, we have variants arising in this coronavirus, and those variants, only one of them right now seems to is not well controlled by the vaccines. So, my advice is, what is my advice? I'm giving it to myself right now. So my granddaughter is in Australia and I have not seen her yet. So she's in her first year of life. And it's one of those things that makes me test my own judgment. But I think the issue being is that if this is a dire emergency, you have to fly. If you're vaccinated, you should go with some confidence, but wear your mask, do your social distancing, wash your hands, and play the game like we've been doing for the last year. I think that's a really important situation. Besides, right now, Australia won't let me in because I'm an American, and our coronavirus level of infections is, just over the top. I would ask that, or would mention that in Australia, near where my son lives, in near Sydney, they have 0 cases of coronavirus infection because they followed all the health recommendations of social distancing, mask wearing, and they're on top of it. When they originally saw cases starting to show up, people went into quarantine right away. So it tells us that those measures work.
That actually was a question that came up. And I don't know who will take this, so I'll let you guys volunteer, as to why we see such differences in COVID spread between various countries around the world. Carl, I think you speak to that a little bit. Sumit or Evan or Anne, do you have any insight into any of that?
I'll let Sumit take that, and then I'll follow up.
Sure. I think it's a multi-layered reason. There were countries like China that could impose severe lockdown measures and stop the spread. More westernized democracies, including South Korea, Australia, Taiwan, also did very well. It was adherence and testing that they got right, which we didn't. We had a major setback in testing from the CDC early on in February, which if we had gotten that right, a lot of it wouldn't have been eliminated, but mitigated. The other interesting thing, which surprised us all, is places like Sub-Saharan Africa and Asia and the third world, the case fatality rates were much lower than here in the US. And so people are pointing to age being in the factor, the US and Western populations having a greater underlying condition and some level of pre-existing immunity. So we know that infection with other coronaviruses can be somewhat protective. So I think all of those together really led to the disparity. I mean, the US has 5% of the world's population and 20% of the cases, right? And we really need to figure out what we did wrong so that never happens again.
I did want to add one thing that bridges both what Sumit said and what Carl said. Even though there's concern about variants emerging that may not be responsive to all of vaccines, that's not an argument not to get vaccinated, because mutations and variations emerge because the virus has an opportunity to propagate. And if a sufficient number of people are vaccinated so that the virus cannot get a foothold, it does not have the opportunity to mutate and for variations to emerge. So the fact that there are variants is not an argument not to be vaccinated. It's actually a very strong argument to get as many people vaccinated as quickly as possible.
And Joe, just to add on that, I think an important point and distinction we need to make is we all are looking through this efficacy number, right? How efficacious is Pfizer versus Moderna versus AstraZeneca? The important point is all of the vaccines are 100% protective against severe disease, regardless of the variant, okay? So you might get the virus with the variant, but you will not die from the virus if you have the vaccine.
That actually, another question from
is our best chance against this virus.
Sorry.
And the vaccine won't kill you either, so.
So there's a lot of rumors out there about the vaccine and things like that. We talked about this before we went on, but I want to ask it for everybody. There was a question about from younger people about does getting the vaccine potentially affect fertility and their ability to have children?
Well, this is an interesting question. We have no evidence that it may affect fertility at this point. But I would say that mothers that are expecting, that have been pregnant, getting the vaccine, I think is something they should do, because when the mother becomes immune, she transfers her immunity to the developing fetus. And that stays with that child when it's born, and if she breastfeeds, her immunity is transferred again through breast milk. And that will provide her protection for as long as she breastfeeds. So I think that is another issue that we should consider about recommending vaccines for pregnant women.
And Anne, as we're seeing from your work too, again, there's some very long-term effects of the disease in our brain and our kidneys, which can have severe issues. So again, we need to get vaccinated to protect ourselves from getting sick at all, because even if we're asymptomatic, we can have this, you can be a long hauler. You can have 20 years from now, something else come up and be an issue, correct?
Yes, definitely, right? Avoid all those long-term consequences by getting vaccinated.
Yeah. We had a question from the audience about the variants, and will new generations of vaccines be based on any of the specific variants that we're seeing?
So right now, Moderna and Pfizer are updating their vaccines and doing trials on the variants. So I want to emphasize that the variants the vaccines are less effective against these variants. They are not ineffective. And so we are making somewhere between 1,000 to 5,000 times more antibody than we need to knock down the virus. So a 20%, 30% drop probably isn't going to move the needle that much. But I anticipate going forward that we will be doing updating the vaccine, and this could be a yearly thing based on the trajectory of the variants that are emerging.
One other thing I'd add to Sumit's comment is that, remember, there's a whole nother feature of your immune system, what we call a cellular immune system, that at this point in time, we have no evidence that would suggest that's been less effective against these variants. So I think that's an important thing to consider. And a fact of being vaccinated, you have this whole other feature of your immune system, the cellular immune system, distinct from the antibodies, can provide protection as well.
I wanted to add one thing too, that our approach to COVID-19 is not unimodal. It's a multimodal approach. So even as we're improving the vaccines and using antibody approaches, we still here at SBP are pursuing drug approaches. So there may be alternative ways for the virus to get into a cell, but there are drugs that can block these alternative routes that can mitigate against some of the other manifestations, including the inflammatory manifestations, the blood vessel manifestations, that would be used in conjunction with the vaccines or immune therapies. So all of that is being pursued here.
Thank you. A lot of questions continue to come in about the vaccine. For people who have had the virus, is there a point in still getting the vaccine, or are they protected from having had the virus?
No, they should get the vaccine. Your immunity will wane. Your natural immunity will wane. So the CDC does recommend, even if you've been infected, you should get the vaccine.
Yeah, that's part of the consequences of having an infection, is that the virus and its effects can continue on. And so the immune system declines, as Sumit mentioned, and the vaccine becomes very important. And it may help protect against the long haulers that Anne's been talking about, that have all the brain and kidney dysfunction.
So interestingly and anecdotally, people who are long haulers who have gotten the vaccine are reporting that their symptoms are being reversed. So we don't know what the underlying basis is, and we're waiting for that data to be borne out. But just another kind of a light at the end of the tunnel for some people suffering from this long hauler syndrome.
And if we need to get vaccines every year, do people need to stay with the same vaccine they've gotten, or does it matter which vaccine they've gotten?
It shouldn't matter what vaccine you've gotten. You should be able to switch out the vaccines. There hasn't been a formal recommendation on this, but from a scientific perspective, each of the vaccines are nearly delivering the identical payload in just a different truck.
Okay. And if you have had the disease, is there a period of time where you should have recovered from it before you get the vaccine?
I think the recommendation is 14 days since you cleared the virus, and also had a previous vaccine of any other kind.
Okay.
The vaccine won't make you sick. I mean, a lot of people think there are side effects, a day or two after you get the vaccine, but in fact, they're not side effects. That's really your immune system at work. And the vaccine is cleared from your body as the immune system takes it and destroys it, and then begins to generate an immune response. So when you feel tired the next day, or a headache, or a little bit of fever and chills, that's your immune system telling you it's working.
It's still uncertain how long the immunity lasts. We're still investigating that, either post-infectious immunity that you develop naturally, or even following the vaccine.
Carl, I want to go back to something you said about expected mothers taking the vaccine and being able to pass that on. Somebody asked if there were any risk of birth defects to the unborn child if a pregnant woman is vaccinated.
I don't think so, because the vaccine doesn't transfer itself into the fetus. So there's no evidence to suggest otherwise, that the vaccine should be very safe from the mother. Again, it's her immune system at work that is driving the protective immunity, and then that's transferred through the umbilical cord right into the fetus, and then again, as I mentioned, through breast milk after the child's born.
Yeah, as a developmental neurobiologist, I can say there is no mechanism where the vaccine would cause any kind of dysgenesis or dysmorphism in a baby.
Sumit, you were going to say something?
Yeah, the only vaccines that are now contraindicated for pregnant women or breastfeeding mothers are live attenuated vaccines, and none of the vaccines that we're using for COVID are of that class. So I would not expect any sort of impact on the developing fetus.
So I want to end with this question, and there are questions we didn't get to, and for those folks, if we didn't get to your question, we will email our scientists and we will get answers, and we will email you with those answers. Sumit, you said that the government should be preparing for the next pandemic. And the question was asked, or what are some of the ways that the government can do that? In your expertise, what are the ways that we can prepare for potentially the next pandemic?
Yeah, I mean, I think, our government, as well as all governments around the world, right? I think there needs to be intergovernmental agencies with more transparent data sharing, preparedness. But in my experience, right, the scientific and medical community came together in ways that I've never seen before in my career, but it was entirely organic. And there was no command and control infrastructure saying, you know, Sumit, you should go screen the known drugs. And Carl, why don't you look at the cytokine response? And Evan, you know, so I think that if there was some level of hierarchical organization, that we prepared these kind of go teams that are at the ready for the next virus, those can be deployed and not have to worry about kind of scientists stepping up and filling out on the niches that they see need to be filled. So I think, you know, we need to go from a bottom-up approach to a top-down approach to have a pandemic preparedness program from a scientific and medical level that can be immune to politics, because that was the other thing that really, I think, derailed a lot of our efforts, was the politicization of the pandemic and health guidance.
Thank you to all of our presenters, and thank you to all of us for joining us. As always, our presentation was recorded and will be emailed to everyone who registered. We've been through a difficult year, but because of the work by hardworking scientists like Drs. Chanda, Snyder, Bang, and Ware, and their colleagues around the world, there's light at the end of this very long tunnel. But our work is not done on COVID-19 or lots of other medical issues that we study here at Sanford Burnham Prebys. You can learn about our other research at future Insights events, and here's a list of those that are upcoming in the next few months. We also want to let you know that thanks to Dinah Rue, who will still be matching COVID-19 donations, you can contribute to the research you heard about today and have that gift matched. Dinah, we are very grateful for your generosity. Thank you. If you would like to learn more about or contribute to any of our research, please contact me at either the email or phone number on your screen. Thank you so much for attending today. Please feel free to share this with your friends and colleagues, and thank you for your interest in our science. Goodbye.